Good afternoon, everyone. It is a pleasure to be here today for giving this webinar. So I'm a researcher at the University of Naples, Federico II in Italy. So I have a computational background in human microbiome data analysis. And so my talk today will be focused on strain-resolved approaches for human microbiome studies. So I want to provide an overview of some of the approaches that can be used for strain-level characterizations, and also try to cover some also of the major challenges that we can have in this field. So the outline of my presentation. So I will start with a very small introduction about microbiome and some metagenomics. So if you are not too familiar with these concepts, then the focus, the main parts of my presentation will be three main parts. So the first is machine learning in metagenomics. So then I will present some tools for strain-resolved metagenomics and also to uncover the hidden diversity from metagenomic data. And finally, something was also more application-oriented, and I will present some examples of studies which I was involved aiming at linking human and food microbiomes. So we start with a very small introduction. So we know the human microbiome, so the heterogenious community of commensal, symbiotic, and pathogenic microorganisms that live in the human body. So when we analyze the human microbiome, so we want to try to answer to multiple questions. So we can summarize as who is there. So we want to try to understand which are the microorganisms present in the microbiome, but also we want to try to understand what they are doing from a functional point of view. So my presentation today, also because it's my major expertise, will be on the bacterial part of the microbiome, and especially the human gut. But of course, it was important to remember that, of course, the approaches that we are seeing today can be also applied to other microbiome body sites, for example, the skin, the vaginal microbiome, the oral microbiome. And of course, there is also the possibility from metagenomics to investigate other, not only the bacterial part, also the viruses, microeukaryotes. So this will be not the focus of my presentation today, but of course it's something that can be achieved from, at least potentially, from metagenomics data. So how can we study the microbiome? Of course, I mean, the study of the microbiome started a long time ago. So in general, so if we see this from a clinical perspective, but of course this can be also applied to other applications. So the traditional way is so getting the sample. Then if you follow a traditional approach of cultivation and isolation, we can cultivate, isolates our microorganisms of interest, and then we can conduct in the lab multiple type of analysis. Of course, with the advent of sequences, we can also sequence the isolates of interest. So analysis of sequences of genomes are done also using the computational approaches. In particular, if we are focusing on this approach, we can usually get the genome, we can reconstruct the genomes, and then we can identify genes of interest. Of course, this is still an approach that is useful and also fundamental in different applications, and can be applied to study only the cultivable microbes. The alternative is the metagenomics, in which we go directly from the sample to the sequencing. In this case, we have a major requirement to do usually extensive computational analysis, since we have a large amount of data and also of different complexity. We can have also some potentialities with respect to the previous approach. We can potentially study unknown microbes. We can reconstruct not genomes of a single organism, but reconstruct potentially multiple genomes from the same sample. And I mean, this is just say the starting point for our analysis. So here I summarize the shotgun metagenomics workflow. This is taken from this nice review paper that I suggest you to have an overview of the workflow to analyze shotgun metagenomics data, so from the start to the end. So these are, I would say, quite complex workflow, but we can summarize it in 5 main steps. So the first is in green here is the experimental pipeline. So basically what we have in the wet lab. So we acquire our sample, we have the phase of DNA extraction, we sequence, and we are getting the metagenome. So usually we are acquiring different samples, hundreds or now thousands or several thousand of samples. We are getting our metagenomic data set that can be very large in terms of size, also several terabytes of data. We have the pre-processing phase in yellow. Usually here we perform 2 main steps. So we filter low quality reads. We perform also identification and removal of host contamination. So now, starting from here, we are considering the pre-processed data set. So it's still large in terms of size. And now we are in the central part of the analysis that can be subdivided into main steps. So in blue is the sequence analysis, primary analysis. Here we have quite usually extensive, intensive, computational pipelines. The objective is to go from the raw data to generate some refined data. We can have a generation of metagenomic assembly, reconstruction of draft genomes, generation of taxonomic profiles, and also functional profiles. Then we have the second part here defined as post-processing phase, in which we want, starting from this refined data, we want to have a proper biological and clinical interpretation. So here we have multiple tasks, multiple objectives, depends of course from our question that we have at the beginning. Here are mainly based on application of biostatistical methods. I mean, here in this figure, I reported some of the major approaches that can be employed. It's also important to consider the last part. You see the validation. So it's also important to try to have a good interpretation of the data. So not just from a statistical point of view, but also have an interaction, of course, with the clinician, the microbiologist. So this is, of course, a very multidisciplinary approach that involves both data analysis and also interpretation of the results. Here is a very summary of what I have already presented, and is like a summary of the workflow from a data science perspective. So we start from our samples, we have the phase of data acquisition, then we have sequences analysis, we have post-processing, and finally we have the validation part. So now I wanted to present a few applications of using machine learning approaches in the metagenomics field. And so this, I mean, one of the questions that we can try to answer is if it can use the microbiome as a predictive tool. So I'm reporting here as a starting point. Actually, the first analysis that I performed in the microbiome field, in which the purpose was to use a machine learning approach to build a predictive modeling. So in this scenario, we want to use the microbiome composition, so in terms of taxonomic composition, as features, and we want to predict the phenotypic characteristics that in this case were the state of disease of the different persons. So here we collected a few public available datasets. So 6 different datasets from different type of diseases, and applied some machine learning approaches, classification approaches, to see if we can distinguish cases from controls. The results were quite, interesting and promising in the sense that for multiple diseases we found a good predictability. So in particular, if we consider the AUC, so this is a value that goes from 0.5 for a random to 1 for a perfect prediction. So here we have some cases in which we have very good predictions, and here we have a medium-low prediction. It was also quite interesting to see if we used some features at the strain level. So these was some, we say not too advanced strain level features, but still that provided a finer resolution with species level features. In half of the cases, we were able to slightly improve the performances. So then also more recently we extended the methodology also, and in particular in this more recent publication, we show how these prediction capabilities are mainly driven by the presence of microbial taxa. So not the predictability is not too related too much on the actual relative abundances values, but is mainly driven on the presence absence of the taxa. This approach can be also important to try to, not just getting numbers in terms of accuracy of classification, but also try to see if there are some signatures of disease in terms of biomarker identification. In particular, putting together the multiple studies. Here are the important scores provided by random forest, but of course there are many other different alternatives. We saw how the presence of some species that were significant that were for the classification, not just in a single study, in a single disease, but they were quite important in multiple diseases. Then, of course, this is possible to perform, also targeted as analysis on more particular disease types. So I'm presenting as an example the case of colorectal cancer. So this was one paper in which I was involved, and in which it was used basically the approach that was proposed in the previous paper. So here, and these are like a framework that beside this particular work is also now quite used also in other studies. And so here, the idea is to perform also a meta-analysis that is able to combine in some way different the findings of different studies to get more robust results. So in this particular case for colorectal cancers, so we started from some available datasets, so 5 different datasets available here. These were acquired from different labs in different countries, and in particular we focus today of the distinction between control and CRC samples. Then there were 2 other studies acquired specifically for this study. So why is important to have the validation on multiple cohorts? As first thing, to identify biomarkers that can be robust across different studies. You can see here, so these are a list of biomarkers enriched in CRC. These are the biomarkers that are more enriched in the control. And we have also the meta-analysis results here. So we can see here, for example, this is the phase of nucleatom that is known to be reached in the CRC, but also a list of other species that are consistently enriched in CRC in multiple population in multiple independent studies. We can also validate the different cohorts in terms of classification accuracy, and we also can have also a validation on independent datasets. So this is the a summary that summarize all the results. We want to try to understand the meaning of this heat map. So here on the diagonal, we have the cross validation results in which we are training and testing on the same data set. So using a cross validation approach, also that is a more common approach. So here we can see how we have some variation of the accuracy that may be due to differences in the population that we have in specific data set, but in general, we have quite a good accuracies. Off of the diagonal, we have instead the cross study predictions. So for example, let's consider this case. It means that we are training on this FANG data set and we are testing on a different data set. So here we are trying to generalize the model on a different data set. In general, we observe a decrease of the accuracy that is in some way expected because we are testing or validating on a different data set. But what is interesting is the numbers reported here using, as we define as leave one data set out analysis. For example, in this case, we are training on all the data sets, but excluding only the data set in which we are testing. For example, in this case, we are training on all different data sets. We are just excluding FANG, that is the data set that is used for testing. So in this case, we can see how, in general, we are reaching very good results that are in some way quite comparable to what we have on the diagonal here. In this case, we see that that adding, integrating samples from multiple datasets can actually improve the general accuracy. So these for me, these are like an example of the analysis that can be also performed on other disease types and other type of disease and also phenotypic characteristics. So we also developed a resource that want also to promote the reuse of the data and to perform meta-analysis or integration on their own generated data with public available data set. So this is the what we called curated metagenomic data resource. So the original publication was published a few years ago, but it is a resource that is continuously updated. And so from our side, there is the downloading and processing of datasets, of public datasets in a uniform way. There is, especially it is also quite a time-consuming effort to try to have the metadata that is standardized. And this package then is available in the bioconductor, and so can be easily downloaded from the IR environment, and this enables to them to perform additional analysis. So to perform, like to apply, for example, a new classification algorithm, some new biostatistical techniques, or also try to integrate public data with newly generated data. So this approach was also used in other papers. I'm just presenting a few examples. So this was a work not in the gut microbiome, but instead on the oral microbiome, in particular the study of the microbiome in periodontitis. We also applied this approach to find microbiome signature associated with in a pediatric allergy cohort, in which we got quite good results. And so this the use of machine learning for predictive modeling is, I would say, also now quite used also, for example, to link diet with microbiome, to link microbiome with diet and other type of data that goes in this direction for personalized nutrition. So in the second part, I want now to focus more on the strain-resolved approaches. So I would say that it is, of course, important to always to remember that in a microbiome, what is can be important are actually the strain. For example, let's consider the case of Escherichia coli, that we know there are commensal, pathogenic, environmental strains that can be very different and have different implications in human health. We say so. Analysis of strains is something quite established if we are working on isolates, and using genomes from isolate sources. This is just one example. For example, if you want to study Staphylococcus strains in a pediatric hospital, we can compare comparative genomic studies. This is something that is becoming, say, more standard also in the metagenomics field, but of course we are still in some way in the phase of developing and improvement of the existing tools. Of course, what we intend with strain-resolved metagenomics, with strain level resolution. So with using 16S, years ago we started from here, so we have maybe a genus level resolution of the microbiome. Using now there is quite established tools for metagenomics that perform species level resolution, so generation of taxonomic profiles species level. Of course, the ongoing step is to perform strain level metagenomic profiling. So we want to define not only which are the species, but also which are the specific strains. There are, a lot of development of different tools in the last few years in this direction, and they are quite used, but of course there is still the room for improvement. Why is it important to perform strain level profiling? So I'm reporting here some applications that can be identification of specific strain, tracking of strains in space and time, and also to open perspectives in terms of genomic and functional characterization. So now I want to present some couple of examples of tools that can be used for strain level metagenomics. So in this field, we can think to two main approaches that are mapping-based versus assembly-based approaches. So I started from the mapping-based approaches. Here, for mapping-based approaches for strain level metagenomics, there are different tools. I'm showing here a couple of examples in tools in which I was involved, and I have used in different applications, but of course there are many other options. So when we do strain level metagenomics, we can think to two main approaches. So the first type of approach starts from the assumption that each strain has a unique combination of genes from the overall species genome. For example, this is the case of Pamphlan. So in general, what is the output that we can get? So this is the output for a single species. This is the case of Escherichia coli. We have here different samples, different metagenomes. Here we have in the different lines the presence absence of specific genes and gene families. So in presence, in red the presence, in yellow the absence. And from here we can identify core genes, genes that are associated with a particular phenotypic characteristic, and so on. An alternative is represented from another approach in which the strain, we start from the assumption that the strain has a unique combination of single nucleotide variants in the core genome. And so this is the case of strain function, in which we are going to identify single nucleotide variants in the core genome. And this is an example of the type of output that you can get. For example, this is the case of Bacterium rectale. Here again, each sample here is a point. And for example, here we can identify major clusters that can be also, then we can also associate with particular phenotypic characteristics. Of course, I would say these are tools that are more tested in the human, I would say, in the human microbiome field, but of course it's also important to try to also to adapt, to test also to more environmental microbiomes. I want to show a few examples on using these tools. For example, using Pamphlan, this is an analysis that we performed a few years ago, in which we focus on, in particular, Prevotella copri, strains in a cohort in which we had groups with different diets. In particular, we had 3 diets, so omnivore, vegetarians, and vegans. And if we are looking at the strains of Prevotella copri, we can see how they differentiate. So we have the omnivores here, the vegetarians, and the vegans. So strain fan, I would say, is more used when we want to do strain tracking and transmissions. I'm just reporting a couple of papers as example. There was the importance to do strain tracking, for example, in mother to infant microbiome transmission, also considering multiple body sites. And I would say in the last years, it also this tool was also applied to perform person-to-person transmission of the microbiome. And of course, these are tools that can be used in any other type of applications that imply the identification of strains across time points or persons. A different, I would say, approach is to try to reconstruct the genomes from our metagenome. And here we need to apply an assembly pipeline. So here is a very schematic representation of what we want to perform during the metagenomic assembly. So we start from the reads. We perform metagenomic assembly. We are getting the contigs. Now we have, with the binning, we want to put together contigs coming from the same microorganism. And so these are the binned contigs. Here we are getting some putative metagenome assembled genomes. We need to perform a quality check to see if these mags are sufficiently complete and not too contaminated. So these are standard metrics for completeness and contamination, say that we used to define medium quality mags. Now from these mags, we can also estimate the taxonomy. In this case, we need also to have an external database with reference genomes. Now we are getting some taxonomy estimated mags and this is a starting point to perform also functional characterization and comparative genomic analysis. This approach was also the base of this paper, in which we perform a large-scale analysis in the human microbiome field, in which we collected, analyzed several thousand of metagenomes. We perform a metagenomic assembly, and we reconstruct a lot of different marks. And we generate a catalog of these marks that we are representing, say, covered, say, all the major species present in the human microbiome. In particular, at that time, we were able to double the set of reference genomes, and we identified around 5,000 different species present at least one human metagenome. So here we considered the mainly stool, but also oral, skin, and vaginal microbiome. And it was also quite interesting to see how a good portion of these species were new, so without isolated genomes, and these species were in particular present in non-westernized populations. This was also interesting and important to improve the mappability of the human microbiome. If we are seeing at this plot, so in red is just using isolated genomes. In blue is also adding the new marks. In this case, we can see how we can have a strong improvement of the mapping rate when we map our raw reads versus the database with all the genes. This approach can be also used in other environments and also to perform integration into different environments. For example, other papers in the human field, but we and also other people investigated also the characterization of the food microbiome. We can also have environmental studies, animal studies, and there is also at this time also to have a standardized taxonomy. And for example, the GTDB probably is represented now. The more standard approach going in this direction. This approach and so this resource can be also used to perform more in-depth analysis for particular taxa, for particular genus or species of interest. This was the case of, for example, Faecalibacterium. So in this case, using thousands of different marks and isolated genomes coming from Faecalibacterium, we identify 22 different genetically distant Faecalibacterium species with 12 species that are prevalent in the human gut, and we found that on average we can have 5 species that occur in the same subject. Then when we link to many different phenotypic characteristics, we found a lot of diversities in terms of age, geographical origin, lifestyle. We saw how Faecalibacterium diversity can be improved by diet, and we also found that it decreases in obesity and inflammatory conditions. And then we also perform an integrative analysis also by identifying hot specific species that we are identifying in different animals. So to conclude, so my presentation, I want also to present a few slides on also my current research. Try to link also the human and the food microbiome, and in particular, so this one of the study that was published was focused on Lactobacillus. In particular, we were interested to assess the prevalence and variability of the lab in the global human gut microbiome. And so for this analysis, so we considered it as a starting point, I mean, similar numbers as already observed in the previous slide, so around 10,000 metagenomes across multiple host conditions. And so we performed a genome-wide analysis by integrating isolated genomes with marks from human and food metagenomes. So in terms of prevalence of lab species, we identified basically 30 species that are quite prevalent in the human gut, in the human microbiome, especially, of course, in the gut. we found important association with the age category, in particular with the lifestyle. So some are enriched in the westernized population, other in the non-westernized populations. Some patterns in terms of geography, and also we found some links with the food microbiome. At this point, it's also important and relevant to perform a more in-depth analysis of specific species doing comparative genomic analysis. This is just an example. This is the case of Streptococcus thermophilus that we found very prevalent in the human gut. And here we integrated food isolates with human isolates, and also food from food, both isolate and mags, from the human only mags, because we didn't find isolates from humans at the time, and we saw a strong overlap of the different strains present in the food and also in the human gut. And now, so we are moving to larger characterization, say, of the food microbiome, and also try to link with the human microbiome. And so this was one of the efforts in the master project funded by the European Commission. And we created a curated food metagenomic data research, in which currently we have more than 2,000 food metagenomes with standardized metadata and process data in terms of taxonomic profiles and also mags. And yeah, we were able to reconstruct more than 10,000 mags that were clustered in more than 1,000 species. And we also identified some species that are new, and we found some overlaps between the food and the human sources. So also extended analysis that not only for the lab, but also for other species of interest. So this concludes my webinar. So I'm here for any questions.
Thank you very much, Eduardo, for a lovely presentation and also for sharing all these resources, which will be very useful for our learners. And thanks everyone for joining today and sending your questions. So we already have quite a few questions in the Q&A box. So I'll be reading these questions, and Eduardo, you can also have a look while I read.
Yeah.
Okay, so the first question is, in terms of sample size, are there guidelines of how many samples or sequencing depth is needed to apply machine learning approaches for metagenomic data?
Yes, so I would say that usually we have at least hundreds of samples. Now there are, I would say, even larger sample sizes, or several thousand of samples. Of course, it depends also on the purpose of your analysis. Of course, if there is the involvement of different, maybe only not only the microbiome, but maybe you are also including other types of variables. Of course, this requires also to increase the sample size. Yeah, I would say in general, at least a few hundreds, but now also with the decreasing of the cost of sequencing, now there are the availability also in the literature of studies with thousands or several thousand of metagenomes. Okay. In terms of sequencing depth, probably, if you are in terms of human microbiome, maybe, it is sufficient to have to have a sufficient sequencing depth to be sure that you can detect at least the majority of your species. Of course, if you are considering maybe other other environments, maybe with a higher also biodiversity, you can also want to increase also the sequencing.
Okay, thank you. How do you account for bias coming from the different studies, sequencing different cohorts, selection of participants, etc.?
Yes, so in general, of course, there are always biases, I would say, because yeah, you give different protocols and so on in different studies. In performing the meta-analysis, what often is performed is to, in some way, to analyze a different data set independently, and then with the meta-analysis, you can have to an integration of the different findings to find robust biomarkers. It's also true that probably you have, the problem of bias is more important if you are comparing also, for example, relative abundances, in which the bias that you have in the protocol, also in the wet lab, can have a strong impact on the relative abundances. For some application which you are interested in doing, for example, strain level analysis or genome comparative genomic analysis, here the bias can be also less. Of course, depends, because at the end, you can have a good identification of for example, of the strains.
Thank you. How do statistical methods and expectations of results differ when using strain resolved versus strain unresolved metagenomics method for health predictions?
So health predictions, so say in general, now most of the analysis is still conducted if you are talking about, for example, predictive model using machine learning, this is usually performed at, for example, species level, okay, because you can find species maybe that are a specific biomarker for a particular disease or a particular type of diet. I would say there is more challenging to perform a strain level analysis in this way to build the models, because probably still unclear, of course, how you need to in some way to group to the different strains to really to go from the strain representation to something that is a feature, because at the end, when you to build a model, at the end you want probably to have features that you can provide as input to your machine learning approach. So, but I want to say this is more challenging, but it probably there is still a lot of research that can be conducted in that direction.
Thank you. How do you account for batch effects when you include or merge multiple data sets and studies? Are there any widely accepted tools for the same?
Okay, there are, accepted guidelines, maybe no. For example, in our analysis, for example, of course, in our analysis, I don't know, for example, if we are considering shotgun data, okay, we are just considering shotgun data with, Illumina sequencing. We are at least removing very, data sets with a very low sequencing depth, or maybe very strange things, or very small sample sizes. Apart from that, probably you cannot do too much. Of course, when you merge different data sets, you need always to have a proper interpretation of the results and see if what is coming out makes sense. So.
What influence do donor, regional, age, sex have on outcome of metagenomic data? And how do you normalize these parameters?
It is, it is not easy to normalize. Of course, also when we find biomarker, for example, particular disease, maybe for, we know that maybe there is an overrepresentation of specific countries, of specific continents. There is a less representation of, for example, of other countries. So of course, there's also something that need to be, for example, checked, verified in other studies. And there is, not a real, you can, of course, for some specific task to do some sort of adjustment of confounders when you do, regression and so on. But there is no, I would say, a general rule on how you should normalize this aspect in general.
Okay. How to match strains from whole genome sequence to ones from metagenomics? Which approach and tool you will suggest?
So when we want to do this comparison, usually we do phylogenetic analysis. So we build phylogenetic trees, and you can see, how much the mags are similar to the genome from isolates, and we say this is a quite robust approach. If you are interested to look at specific presence on the absence of specific genes, I would say this is more challenging, because from for example, from the mags, you know that you can have contamination and so on. So this can have an impact on the quality of your mags, and so this is more challenging when you want to compare with an isolated genome.
Okay, thank you. Next question, I think you partly answered it already, but I'll just go through it again. What's your opinion in meta-analysis approach, such as effects from batch, experimental, and technical interstudy, as these data are from different sequencing platforms? How to handle these challenges during machine learning model constructions? And there's another part of this question. Also, your opinion on integration of host-associated metadata, such as sex, age, geographical location, diets, antibiotic consumption, etc. How can we integrate into the machine learning model construction?
Yes. In the meta-analysis approach, we identify the biomarkers independently from each data set. Then we found the robust biomarkers in the meta-analysis. For the machine learning, from our experience, what is important is to do what we define the cross study validation, in which you try to train in one data set and test on another data set. And usually, you can really in some way test if your signal that you have in your training data is also transferable to another one. Of course, if you have a different data set with very different characteristics in terms of population, one with a very old age, another with a very young population, or at the end is I would say is a limitation, I would say, of your analysis of your experimental data. And so you can in some way adjust in the statistical part, but of course at the end is a limitation of what you can get.
Thank you. And next is about metagenomics data. Which database would you recommend for taxonomic identification in 16S sequencing?
So there are different databases for 16S. You can I mean, I cannot tell you which is the best one based on my for the bacterial nucleatum, because it's not something that I have really experience in that.
Yeah, thank you. Yeah, I can just add, because I'm from EMBL-EBI, so we have a resource called Magnify, so you can get lots of data there for metagenomes. Okay, would it be adequate to use the strain FLAN tool for a study of bacterial transmission from the environment to humans?
When do you suggest to perform the genomic assembly? Okay, so yeah, for the metagenomic assembly, okay, so metagenomic assembly is the de novo assembly, so it is not biased towards non-species. For example, is essential or very important also in less reconstructed less characterized environments. For example, if you are not focused on the human, but for example in other animal microbiomes, environmental microbiomes, so we have a lot of say hidden diversity. So in that for that application is anyhow important to perform metagenomic assembly, also because probably you can get a lot of marks from new species. So for the human, I would say that specific thing is less important, since we have probably now a very good database with that covers say the majority of the tags in a very good way. So in that case, with strain FLAN and in Papa, you can have more accuracy, I would say, if you want to detect the strains. Okay. Particular strain FLAN, I would say the strain approach is the optimal one if you want to track strains. PanFLAN instead, if you want to do also functional analysis, because it provides you the presence and absence of specific genes, and you can do functional analysis based on that.
Okay, next question. Are these data sets, I think the ones which you presented, are available to anyone to use? Is the metagenome sequencing all from saliva samples, or there are some which are from fecal samples?
So basically all the data that I presented are public available. I would say the majority of my presentation was on stool samples, but yeah, there are of course also a lot of studies on the oral microbiome, including also the saliva samples. Yeah, and yeah, for example, also in the curated metagenomic data research, you can, if you are interested in fecal or saliva sample, you can find data sets available with also the metadata. Can be like a starting point if you are interested to do some analysis.
Okay, so next one I can take is thank you very much for presentation. More from a data science perspective, this hard approach is very refreshing. How far do you think we are from a Star Trek kind of fiction in which we swab an ocean, put into a portable machine, and quickly get a full profile of species and strains? And how do these bacteria feel, etc.? Do you think there is a potential for systemic, statistical, and biological discoveries that can provide this? Or do you think such complexity will always be a significant limit, and the field will continue evolve past photonics, quantum computers, ChatGPT, etc.?
Considering I would say more recent technological developments, of course, there was the idea, I would say, that should try to as you are saying, to have a portable machines, very devices in which you can do a sort of live sequencing and analysis. We are still not there. I mean, so we'll see in the future. I mean, if this will happen or not. So not I mean, not now. But.
Right. Thank you. So this is the last question we can take. We just have 1 more minute. Thank you for your interesting presentation. What would be a realistic time frame for such an analysis like you presented once you collected your data sets?
So of course, I mean, it depends on the complexity of the analysis, for the expertise that you are in the data analysis. I mean, the sample size. I mean, for example, in the metagenomics, so the metagenomic data are quite so big. If you have, I don't know, hundreds of thousands, you use a bottleneck also in the computation. I mean, if you want to generate, all the genomes, the taxonomic profiles, functional profiles, strain level profiles, it also can take several weeks, depending also on your computational resources. Starting from there, okay, if you have all the data ready, the metadata ready, that is actually quite important aspect, because most of the time metadata is not, you spend a lot of time to clean the metadata, collecting, getting missing data, integrating new variables. If you have everything ready, I mean, then it can take a few weeks to perform all the statistical analysis if you have of course the experience in doing that.